Bismillahirrahmanirrahim. In the previous lecture, we start discussion on classification of antibiotics. And in the previous lecture, I discussed beta-lactam antibiotics, all the classes of cell wall synthesis inhibitor, including beta-lactam antibiotics and their mechanism of action. Now, in this lecture, we will discuss the other classes of antibiotics. As I mentioned previously, there are five major classes of antibiotic according to their mechanism of action. We have already discussed cell wall synthesis inhibitor. Now we will discuss the remaining four classes, that is, agents that interfere with cell membrane integrity, then protein synthesis inhibitor, nucleic acid synthesis inhibitor, and agents that interfere with folic acid metabolism or antimetabolites. So first of all, we have already discussed the cell wall synthesis inhibitor. Now the second category, it includes those agents that act on the cell membrane. Now first, the cell membrane of all the microorganisms, whether it's prokaryote or eukaryote, it is made up of phospholipids and it carries negative charge on it. Now, these cell membrane integrity inhibitor, it include polymyxin, like polymyxin B, polymyxin E. So these polymyxins, they have a strong positive charge on them. They are cationic in nature. So being cationic in nature, they have a natural tendency to interact strongly with the negatively charged phospholipid membrane. So it adhere very firmly to the cell membrane because of opposite charges, and it basically it causes the calcium and magnesium ions to ooze out from the cell membrane, and so it causes the destabilization of the outer membrane, and because of this destabilization, it causes pore formation in the cell membrane, and the cellular content, intracellular cell content, it oozes out from inside to the outside. So, because its primary target is LPS, so definitely all the polymyxins, they target only gram-negative bacteria, and they are bactericidal in nature. I mean, they kill the bacteria. So that's all about polymyxin or cell membrane integrity inhibitors. So you see, they causes destabilization of cell membrane because of opposite charges interaction. So next category of antibiotic is protein synthesis inhibitor. So because before going into details of protein synthesis inhibitors in bacteria, it is necessary to understand the process of translation or protein synthesis in bacteria. In bacteria, as you know, ribosomes are responsible for protein synthesis in both prokaryotes and eukaryotes. However, the ribosome of prokaryote is different from the ribosome of eukaryotes. In prokaryotes, it is 70S, consisting of 50S larger subunit and 30S smaller subunit. Whereas in eukaryote, you know that it's 80S, consisting of 60S larger subunit and 40S smaller subunit. So the protein synthetic machinery, it includes ribosome and of course the messenger RNA that is transported from the nucleus to the cytoplasm. Now, in the cytoplasm, there is messenger RNA, there are ribosomes as well. Now, when the translation has to be started, first of all, the initiation complexes, initiation protein IF1 and IF3, these are the main initiation protein. They bind to 30S ribosomal subunit, and it got separated from the ribosomal complex. Smaller subunit, it then move toward the messenger RNA and get strongly attached at the Shine Delgarno sequence. This red one is the Shine Delgarno sequence. Now, actually. The 30S ribosomal RNA has 30S ribosomal subunit has sequence or known as 16S ribosomal RNA that is complementary to Shine-Dalgarno sequence. So it binds firmly to the Shine-Dalgarno sequence, and the objective, and it is upstream the actual start codon. The purpose of this binding is to align ribosome at the specific site, at the you can say at the appropriate site from where the translation can be started. So after the binding of a smaller subunit to the messenger RNA, another initiation complex called IF2, it binds to transfer RNA carrying the anticodon to the start codon, and then it binds to the start codon on the messenger RNA. So you can see, okay, now the initiation complex is complete. The start codon has its anticodon, and then after its binding, now the larger subunit of ribosome, that is 50S ribosomal RNA, 50S ribosomal subunit, it got attracted to this complex, and it got fixed at its place. Now ribosome has actually three sides. It's not demarcated, but according to their designated function, you can divide it into three. You can say three different sites: A site, P site, and E site. Now, A site of ribosome is for the entry of new transfer RNA. P is for a peptidyl chain, and E is the exit site. Now you can see now this transfer RNA that is that carries the anticodon of the start codon, it bind at the P site. Initially it binds at the P site, and A site and E site both are empty. Now the transfer RNA, transfer RNA, it brings the anticodon to this codon present on the messenger RNA. So you can see next codon is U. So the anticodon should be A. Now the transfer RNA carrying the amino acid will enter the A site. If the transfer RNA carrying the amino acid are complementary to this codon, then it will bind here, otherwise it will eject it out. So you can see this transfer RNA complementary to this codon has bind here. Now the first codon, first transfer RNA is bind at the start codon, and next transfer RNA at the next codon. So now when these two places are occupied, A and P sites are occupied, then there a peptide linkage established between these two amino acids. So as the peptide linkage is formed between these two amino acids, the ribosome, it moves one step here. So this is the result of moving a ribosome to a step forward. So as a result of this, now the peptide chain would be at P position, and and the transfer RNA at E site. It has already, because donated its amino acid at the P site. So it's now empty, and it will be released. And now the A site is also empty, and now the new transfer RNA will come here. And if the sequence of the transfer RNA would be G के complementary would be C, then C, If the next transfer RNA carrying CCG will come here, it will bind to this codon. Otherwise, it will be released from here. So that's how when the transfer RNA carrying anticodon to this codon will come here, it will bind here, and then again the peptide linkage between this chain and the new peptide will get established. And as the peptide link will establish, ribosome will move a step step forward. So this transfer RNA will be released from the E side. So that's how the peptide chain is formed, and ultimately the stop codon is reached. And when the stop codon is reached, then because there is no anticodon to stop codon, so the growth of the peptide chain would get stopped, and that's how the polypeptide chain is formed in during translation step. Now protein synthesis inhibitor, what they do, they halt this process at various steps. Like a few of the antibiotics, they intercept at 30S ribosomal subunit at 30S stage, and a few of them they inhibit the protein synthesis at 50S subunit. Tetracycline and aminoglycosides, they are very well-known antibiotics. Tetracycline, it binds to 30S ribosomal subunit and interfere with binding of transfer RNA to ribosomal complex. ठीक है, यानी which is the very first step. Interfere with the binding of transfer RNA to ribosomal, mean it intervene the translation process at the A site, जहाँ पे transfer RNA it enters and it binds to the codon. So it interfere with the protein synthesis at the very first step, and then aminoglycoside, it also bind to 30S and causes mRNA codon to be misread. Definitely when aminoglycoside, it will bind to the codon of messenger RNA. Definitely the space would be occupied by the antibiotic, and the codon will be misread. Thus it interfere with the initiation complex of 30S and 50S with messenger RNA. So both tetracycline and aminoglycoside, they interfere or intervene in the translation process at 30S ribosomal subunit, whereas linezolid, chloramphenicol, and macrolides, they interfere or inhibit protein synthesis at the 50S ribosomal subunit. Now, because you have understood completely the whole process of translation, so you can easily understand how these antibiotic linezolid, chloramphenicol, and macrolide, they are inhibiting translation in bacteria. That's all about protein synthesis inhibitor. Now we will move to the next class of antibiotic, which is the nucleic acid synthesis inhibitor. Nucleic acid synthesis inhibitor are divided into those antibiotics that inhibit DNA synthesis, and it also include those antibiotic which inhibit or interfere with RNA synthesis. Okay, so basically, the first step during DNA replication or transcription is, because DNA is very much super quiet, and when DNA has to replicate itself, or when the DNA is subjected to transcription, first it has to unwind, to lose the sequence of DNA. So there are two enzymes, DNA gyrases and topoisomerases, that plays a vital role in you can say they introduce a nick in or a small break in the DNA, a single strand of DNA, and thus the supercoiling is released. When the supercoiling is released, it's easier to read the codon present in the DNA strands. So basically, nucleic acid synthesis inhibitor, they actually interfere with the working of DNA gyrases or topoisomerases, and thus they they actually intervene the process of unwinding, or they introduce instead of introducing the single strand neck, they causes the break in double strand. So that's how they interfere with the normal cycle of replication or transcription. So that's all about nucleic acid synthesis inhibitor, and it include because the nucleic acid synthesis inhibitor, it include quinolones and fluoroquinolones. Ciprofloxacin is the very common examples of quinolone antibiotics, and RNA synthesis inhibitors, it include rifamycin. Rifamycin actually it binds to DNA dependent RNA polymerase. So it interfere with the transcription process. So that's how the nucleic acid synthesis inhibitor they work. Now the last class of antibiotic is folic acid synthesis inhibitor. Actually, bacteria can't get as in human or eukaryotes. We actually consume folic acid from the food. In case of bacteria, bacteria can't take folic acid from the food. It has to synthesize inside its body. So folic acid is synthesized from the two precursor molecule known as pyridine and PABA or pyraminobenzoic acid. Now, when these two moieties they are combined, with the help of enzyme known as dihydropteroate synthetase, and they form dihydrofolic acid. Dihydrofolic acid is reduced to tetrahydrofolic acid with the help of enzyme known as dihydrofolate reductase. And now the tetrahydrofolic acid is the active form of actually folic acid that leads to the formation of purines. And purines, DNA is made up of purine and pyrimidine. So when the purine synthesis would be halted, the whole bacterial cycle would be disturbed or inhibited. So basically, folic acid synthesis inhibitor, what they do, there are two categories of folic acid synthesis inhibitor. First one is sulfonamides. Sulfonamides, they are structural analog of PABA. They have a very close symmetry to paraminobenzoic acid. So with dihydropteroate synthetase, instead of taking the PABA, sulfonamides occupy the pockets of that enzyme, and the first step of folic acid synthesis is thus inhibited. Whereas in case of trimethoprim, what they do, they actually inhibit dihydrofolate reductase, or they halt or they interfere with the second process of folic acid synthesis. And usually folic acid synthesis inhibitor, they are used in combination. Trimethoprim and sulfonamides, they are used in combination, so that to increase their efficacy. Of course, sulfonamide will inhibit the synthesis at first step, and trimethoprim will inhibit the synthesis at the last step. And when they both act together, the whole folic acid metabolism would be halted, and the DNA replication would get stopped. So that's all about different classes of antibiotic and their mechanism of action. So just a brief overview. Antibiotics are broadly classified into 5 different groups according to their mechanism of action. Cell wall synthesis inhibitor, they act on the bacterial cell wall, then cell membrane agents that interfere with the cell membrane, then protein synthesis inhibitor, nucleic acid synthesis inhibitor, and then antimetabolite. So all these different categories of antibiotic, they act on different targets and they kill the bacteria. We have discussed in detail all these different categories of antibiotic, how they actually how they form complexes with the bacteria and causes the bacteria to stop its replication or growth. Few of the agents are bactericidal and many are bacteriostatic as well. So So that's all about antibiotic. In the next lecture, inshallah, we will discuss how the bacteria develop the survival strategy to counter these antibiotics. So that's all for today.